So we're going to get going here. Everybody, welcome to day 2 of our rare rounds and our repurposing for rare disease session. I'm going to step out of kind of presentation mode here for a couple of housekeeping items, but we will be. I just want to make sure that I've got everybody's that everybody is understanding the tools and everything. So thank you so much for joining us. Before we get kicked off, just want to remind everyone that all attendees are currently muted by me, the moderator, Christian Rubio, to reduce any interruptions. We encourage you to use the chat function in the Zoom in the Zoom window for any questions you may have throughout the session. We will present them to the speakers as they come in, or we'll actually probably get through some slides first and then open it up to discussion. To open the chat, please click the chat button located at the bottom or in the meeting controls of your screen near the mute icon. If there are any questions that are not able to be addressed by the speakers or we run short on time, we'll do our best to follow up after the program. In the interest of making us all feel more connected, I'd like to ask everyone to actually, depending on the size of the group, we'll decide if we want to turn on the videos or not. But that will be after some presentations here. So I'm now pleased to welcome our discussion leaders, David Feigenbaum from University of Pennsylvania Orphan Disease Center and the Castleman Disease Collaborative Network, and Bruce Bloom and from Helix as Chief Collaboration Officer and founder of Cures Within Reach. They're each going to share a little bit more complete background as we delve into the conversation, but for now, I'd like to invite David to get us going and kick things off. So David, the floor is yours.
Thanks so much, Christian. And thank you to everyone who has called in today. I really enjoyed having the opportunity to present along with Bruce yesterday and really pleased to have this opportunity again today. So we'll be talking about drug repurposing in rare diseases, and something we mentioned yesterday just to make sure we're all on the same page, because repurposing can have multiple definitions, but when we're talking about drug repurposing, we're really talking about taking a drug that was initially developed for one condition and trying it in a new condition. It might be that that drug is already FDA approved for the particular condition, or it may be that the drug was never approved for any condition. Both of those are possible, and it may be that you go on to get this new drug or this drug approved for a new rare disease, or it may be that you don't get it approved, but you demonstrate that it's actually effective, and both of which are obviously certainly important in situations like this. And also to further frame why Bruce and I are talking to you today, there are 7,000 rare diseases, almost all of which do not have an FDA-approved therapy, and there are about 1,500 drugs that are already FDA approved. And so the hope is that we can match all those diseases without any treatments with the disease or with the drugs that already exist. So today I'll be sharing my experiences from 5 perspectives. That as a caregiver of a patient my mom who passed away from brain cancer while I was in college, and a number of off-label drugs were tried and repurposed for her brain cancer, unfortunately unsuccessfully. That inspired me to want to go into medicine. And then during my 3rd year of medical school, I became deathly ill with a disease called idiopathic multicentric Castleman disease, a hyperinflammatory disorder that made me so sick that I had my last rites read to me, and unfortunately went on to have 4 more deadly relapses. Having this illness and learning that there were no options for me inspired me to decide to dedicate my life towards research into Castleman disease, and eventually identified a drug, sirolimus, that had been developed 30 years before for a totally separate condition that we decided to repurpose, and I've been on now for the last 6 years. And then my last perspective is as an advocate. I run a foundation called the Castleman Disease Collaborative Network. We spend a lot of our effort thinking about ways to accelerate drug repurposing for Castleman disease, but also beyond Castleman disease. So I thought I would start out by sharing our approach to drug repurposing. When I think about our approach, I really break it into 5 steps. And this is not the only approach to drug repurposing. You're going to hear about another approach from Bruce, and we'll even talk about additional approaches probably during the Q&A. But the approach that we take is to start out by first really interrogating disease biology, trying to understand exactly what is happening in samples from humans with that given rare disease, from actual patients, not just animals, but what's going on in patients. Next, once we better understand what's happening in these cells, we then ask the question, what drugs already exist that are known to either decrease what's too high or increase what's too low? Are there things that maybe we could perturb that could actually turn around the disease? Once we have those candidates, we move forward to in vitro or in vivo testing. In vitro means that we test the drug on cells in a dish. In vivo means we test the drug on animals that are animal models of the disease, so live versus in a dish. And then once we have identified that certain drugs have a high likelihood of having an effect in Castleman disease, we publish that information in peer-reviewed journals. And because Castleman disease is so deadly and so few options exist, clinicians will actually begin to try these drugs in patients. And sometimes these drugs work, sometimes they don't, but we always systematically track how well those drugs perform in patients. And then based on those systematic evaluations, we move forward the most promising drugs to clinical trials. The clinical trial then tries to assess how well does the drug work, so then we can change practice and change the way that Castleman's patients are treated worldwide. So I thought I would take us through 3 examples of this path. The first was work that we did in my lab that identified a particular communication line called the mTOR pathway to be highly activated. We published that data and then actually, based on that information, decided to start testing the drug on me. We kind of skipped testing it in cell lines or animal models because frankly, I was running out of time and decided to try the drug on myself. This drug was reported in the New York Times as a headline, "Doctor Cure Thyself," which I think is a bit of an overstatement. I think that it's doctor helping himself a little bit for right now and hopefully for a lot longer. But thankfully, that helped to get the word out about this approach. And as a result, a number of other patients have been treated with the exact same drug, sirolimus. And thankfully, it has worked for about 1/3 of patients. Unfortunately, it's not working for 2/3 of us. But based on the data we generated and based on the experience of it helping about 1/3 of patients, that was enough to have us get a grant from the federal government to launch a clinical trial that's currently enrolling patients. The next question that I, of course, asked after we started treating me with this drug was, well, how many other patients could this drug potentially help? So we did a series of experiments where we found that particular communication line that was active for me is also active for other patients. We moved forward into in vitro testing where we tested out this molecule and a couple others that have a similar mechanism of action. And we generated some really promising results. We run a natural history registry called Accelerate, where we keep track of all of the patients worldwide with Castleman disease, what treatments they get, how well they do, how well they don't do, to really understand what's happening in the natural history of this disease. And based on this work, we've identified a new drug target called the JAK1/2 signaling pathway. Thankfully, there are drugs that already exist that can target JAK1 and 2. So as a result of this pathway, we've got a drug that's saving my life. I'm literally here today because of this drug that I identified, but also identified another drug that seems very promising that will hopefully be studied in trials soon. When the coronavirus pandemic hit the US in early March, my lab and I decided that we wanted to try to apply a similar approach from what we've been doing for Castleman disease towards COVID-19. And we launched a program called the Corona Project. It's the world's only project that's keeping track of all drugs that have been used against patients with COVID-19. So we've now captured data on over 160 different drugs that have been tried thus far against COVID-19. We've heard about a handful of those in the media, but actually there were more than 160 drugs that have been tried. We published our results from the first 9,000 patients a few weeks ago, and now we're up to over 20,000 patients worth of data. In parallel, we're doing a similar sort of what we call immunophenotyping, meaning we're characterizing the immune system of patients with COVID-19, looking for similarities and differences between Castleman's and COVID, because there's a lot of overlap. And based on our work, we've identified sirolimus and this JAK1/2 inhibitor as candidates that could potentially be effective against COVID-19. So I thought it would be helpful to share these kind of steps that we go through along with examples of work that we've done. And before I pass it on to Bruce, just want to close by sharing that I tried to document and really chronicle this whole pathway and process through a book I wrote that came out last fall called Chasing My Cure. And I'll be sharing during the closing remarks today that I'm delighted to be able to share copies of Chasing My Cure. If you want to send an email to me, I'll be able to send out about 100 copies. So send me an email and your mailing address if you'd like a copy of Chasing My Cure. It really tries to delineate the steps that we followed in order to identify a drug that saved my life. So now I'd like to pass it on to Bruce.
Thanks, David.
Thank you, David.
Thank you.
I'm going to turn on my screen. And it's also great to be here with David. It was the first time he and I got to sort of co-moderate any kind of a presentation. We've known each other for a long time, and we actually first connected not around drug repurposing, but around technology when he was back at Penn in his early days of training. I'm also glad to see, attending today are some people that I just want to do shout out to. So Barbara Goodman, the president and chief operating officer of Cures Within Reach, they had a phenomenal event yesterday afternoon with 5 really powerful repurposing projects, at least one of which got funded. And Brian Frasca, who's a good friend and great support for Cures Within Reach, and a new friend of mine that I've actually just met. So Eric is on the phone as well. So welcome those of you who I don't know. I'm eager to get to know you and to hear some of your questions. So I want to just talk a little bit about my background. I'm actually an attorney and a dentist so I have kind of a strange background for getting into this work, but early in the 80s, I started to do some clinical research in the consumer products and other kinds of sort of claims research area. And I really liked supervising clinical research. And after some other kinds of things in the medical malpractice and consumer products space, I ended up at a not-for-profit called Goldman Philanthropic Partnerships, which in 2005 we converted into a public charity that is now called Cures Within Reach that Barbara is running. And during the time we were there, we sort of fell into repurposing, and more than a dozen therapies have reached patients, many of which are similar to how David said that, we end up with things that are used off-label and, for which we're able to get physicians to use them. I'm now the Chief Collaboration Officer at Helix, which is a biotech that uses artificial intelligence and repurposing to find new therapies for rare diseases. And I'm lucky to have a global network of really smart people who do all the hard work, and my job is to sort of bring them all together so that they can make an impact in rare disease patients. So I want to talk to you about how we do things at Helix, and it's a little bit different than the way that David does things in his space. And the real difference is instead of using our own knowledge about disease, where we go in and do a lot of research or have a lot of knowledge about a particular disease, we gather all the data from pretty much all the medical and scientific literature and lots of other data, and we put it all in one place and let the computer use its training to figure out how to connect diseases on one side with drugs on the other side. And in between, it looks at billions of connections between data points that are in published literature, which you see at the bottom of my screen here, this unstructured data that the computer reads and uses natural language processing to find connections between words that make sense. So if there was the Castleman's disease was in there and sirolimus was in there, it would look to see, is this drug related to this disease in any way? And if so, does it treat the disease? Does it cause the disease? Is it not related to the disease at all? It has to think of the context of the information in between sirolimus and Castleman's disease. And so, but it gives us a tremendous amount of information. We also have at the top of the slide, you see this structured data. Those are things like Drug Bank and Kemble and other places where really highly curated data has been put in by many, many scientists, and it's growing every day. And these talk about drug structures and drug activities and disease activities. And the more of that data we have, the more of these points in between where they can all come together. And then in between, we have our own proprietary data that we either generate through getting patient samples or cellular lines or some animal models, or we in-license it from really smart physicians, laboratories, academics, CROs. And we put all that stuff together, and then we ask the artificial intelligence questions very much the same as David would ask his team members and other scientists, the same kinds of questions. And what we're trying to tease out is non-obvious connections between a disease and a drug. So the obvious ones we can get to, so David said sirolimus works on cytokine storms. And so when you realize COVID, some of the aspects of COVID are caused by cytokine storms, it's obvious to think about how those might work. But there are lots of things that are not obvious from that. And this system tries to make those connections, and it uses artificial intelligent reasoning to get there. And then we get these predictions, and we get long lists of predictions, and then our human scientists look at these, much in the way David and his team look once they've figured out the biology. And we think about, does it really make sense, this particular drug connected to this disease? Does that make sense, all the evidence in between? The other interesting thing that the AI does is if there's strong evidence almost to connect them, but there's a missing piece, the artificial intelligence will predict what that missing piece might be and allow the connection to go through. And it says, I made a guess here, think of some science that would validate that. But what we end up with is a whole long list of monotherapy, drug repurposing that might help a particular disease, also some combinations that might help those disease. And along the way, we might generate some ideas around biomarkers or other things that could be used, both preclinically and clinically, to validate whether a particular drug therapy is actually helping that particular disease. So the last thing I want to share, like David did, is sort of an example of this. So this is an example in Fragile X. So there are Fragile X mice that recapitulate the disease. And we found, after doing all that work, about 18 different therapies we thought were safe enough to give to patients, that didn't have overlapping mechanisms of action, that didn't have drug interactions with the other drugs. And we thought, all right, let's test these in the animal model. And 17 out of 18 rescued at least one behavior that's indicative of Fragile X. But as we keep going along, 75% almost rescued 3 or more behaviors. And I don't know if you can see it because I can't figure out how to get all the pictures off the screen. You guys can see that last piece? Okay, so, 50% rescued all 4 behaviors. And of those 50%, we're now getting ready to test in the clinic, those on live patients. So, and none of these, of the 18 drugs, none of these had ever been used in Fragile X before. And there wasn't direct, obvious connection between maybe a related disease, another autistic-like disorder. These are all sort of novel things that you might not have figured out had it not been for the AI. And so the last thing is that, the timeline between when we started this in the first experimental validation and when we're getting ready to do a clinical trial is pretty short. The prediction time is really short to get us right into in vitro and in vivo validation. So I'll stop there. The rest of it is like David, make sure you move it along. If it looks like it has commercial value, we'll take it to clinical trial. If it doesn't, we'll publish the results, let physicians use it off label, see if we can get it to patients like that. So Christian, back to you.
Thank you. We are going to...
I'm going to stop presenting. If I can figure out how to do that. So you can have, can you take it back?
Sure. Yeah. I will take it back. Actually, I'm not going to put up slides so that I can keep reading the questions that come in. But we are going to get into some questions here. So what are some of the potential pitfalls or challenges in the process of repurposing, David? I think that was one that was top line.
Sure. I'll start with it. I think that a major pitfall to consider is that there are oftentimes drugs that we get really excited about based on the lab research we do and based on our in vitro and in vivo experiments that don't oftentimes result in solutions for patients. And so I think that it's important to remember a couple things. One, I think that you need multiple shots on goal. Just because you have one drug that looks promising in mice does not mean that that drug's necessarily going to help patients. However, if you don't have any drugs that are promising in mice, you most certainly don't have something that's going to be promising for patients. So having a drug that works in mice is better than not having a drug that works in mice. But I think that it's important to remember that that drug may not work in humans. And so continuing to be relentless and continuing to push for maybe new animal models that maybe even better recapitulate human disease. At the end of the day, we generate what we call animal models, and those are really just animals that have features that look like a human disease that we care about, and we try to cure those animal diseases. It's great when you can have many different animal models of the same disease, and if the drug works on different models, that makes you feel even better. But at the end of the day, I think that drug repurposing, and I'm preaching to the choir, you guys know this all too well, drug repurposing and rare disease drug development is really challenging, but it can be done. These are paths that Bruce talked about doing this with Cure Accelerator 14 times. We've done this successfully once, and we've got a couple more coming down the pipeline that we're excited about. So it can be done, but it's important to remember that you need multiple shots on goal.
Yeah. I'll say probably, Christian, the biggest challenge is that for a lot of drug repurposing, there's no economic incentive for it. David repurposed sirolimus. Interestingly, at Cures Within Reach, we had some scientists that repurposed sirolimus for a number of pediatric autoimmune cytopenias, much the same kind of thing, life-saving results. And but diseases of 600 to 1,000 people in the United States, nobody's ever going to get their return on investment back in doing all the phase 2 and 3 clinical trials, even if you can accumulate enough patients to do that. And so we have to figure out other ways of getting drug repurposing to patients, whether it's the kind of work that David does in publishing the results, or looking at new economic incentives that could be put in place, using philanthropy to pay for some of these things. There's all sorts of ways that companies are looking at this. Maybe the drug isn't profitable, but there's a diagnostic test that a patient group needs to take to decide if they're actually going to benefit from it and whether the therapy's working. And so you bring the drug to market and you make your money on the diagnostics. So there's all sorts of things that people are thinking about, but maybe that's the biggest hurdle that I think about. The other thing I would say here, and this has changed in the last 4 months, but up until, I would say, the end of February of this year, those of us in the drug repurposing space think it's one of the smartest ways to create therapies. But for everybody else in the new chemical entity space, it seems like nothing worth using. And the COVID issue has brought to light, I think a lot of lay people know the term drug repurposing because of what's happened with this. And I hope that it shifts the thinking on the value of repurposing drugs. And that the other neat thing is, I mean, at Helix, we have 4 different ways that we evaluate our AI, but we've seen dozens of other ways that other companies are evaluating, data to predict drugs. And so, and people are just sharing, wildly on this. So I think there's going to be a huge improvement in how we do drug repurposing from all of the kinds of things that we see, how we structure clinical trials, how we use AI, how we collaborate. That's been another really great thing that we've seen here. I mean, labs that wouldn't talk to each other because they're competing for a grant, suddenly working together and sharing data it's been really great.
I totally agree, Bruce. I think that there's some really nice silver linings. COVID-19 has been unequivocally awful in every way, but I do think there are some silver linings that come from this, just as you mentioned. And yeah, it's kind of, I think one of the reasons that people look at drug repurposing for COVID is that we as a medical community and as a society, we're kind of backed against the wall. We have this awful disease that's killing a proportion of patients, and no new drug could be developed in 2 months' time. That's just impossible. So we've been kind of forced into drug repurposing. Well, I think most of us on this call, you included, me included, would say that in the rare disease space, we've been forced into drug repurposing for a long time, because many of our diseases don't have commercial interest, and many of our diseases, we don't have time to wait. I mean, we all have urgency. We need drugs. And so I think that this sort of urgency has been felt beyond rare disease, and I'm hopeful that that this will be contagious and that it will have an impact in rare disease.
Well, and David, you know, you and other physicians do drug repurposing all the time. When you are presented with a patient and you don't have anything for them, you start thinking, well, is there potential that some drug that's already available could moderate their condition in some way? I know when I was at Cures Within Reach years ago, we looked up one year, and it was something like 21% of all prescriptions written in the United States were written off-label. In other words, drug repurposing N of 1 trial happening in a physician's office to try and find something that could help a patient. And some of those lead to larger bits of information so that it can be more widely used, but a lot of it just stays in that clinical practice for that particular patient or group of patients.
Yeah, which I think is probably one of the biggest, another pitfall to answer Christian's earlier question is just that we don't do a great job of systematically tracking when drugs are used off label. And so unfortunately, that means if the drug is not working and it keeps not working and you keep giving it to people, N equals 1, that's bad. And if it's working and you don't know to give it to other people, that's also bad, because we need to get that information out. So for Castleman's, we've tried to solve that, but obviously there's a lot more or many more diseases where that needs to be solved. I also wanted to highlight, Bruce, something you and I were talking about earlier, and that's that the two approaches that we've shared are not the only two approaches to drug repurposing. There's another approach, which Bruce mentioned, which is the clinician in her office thinking, this is an interesting disease, and I've used this drug before, and you know, making, you know, connecting the dots there. That's one. You know, that's another way that drug repurposing happens. And at the end of the day, if it helps patients, you know, that's what we need. Another way of drug repurposing is through high throughput drug screening, and that's where you get full libraries of chemical compounds, typically all FDA approved drugs, plus a number of drugs that have not yet been FDA approved, but that we know exist and that have already been developed to some stage. And you test those compounds on cells from either an animal model of the disease or directly from patients. And that's a really exciting approach, because you're actually testing the drug on cells, and you're not just testing a few. Like in my lab, we find the highest candidates, the best candidates, and we test a few, and then we move forward. This is a way to say in an unbiased way, I want to look at everything. The limitation for a lot of rare diseases is knowing exactly what do you want to test in that dish. You might put a drug on cells, but what are you looking for to happen? The cells are not going to say, you did it, that you fixed me. You're going to have to measure something. And unfortunately, what you measure isn't always a perfect reflection around what's going to actually happen in the human body. So it's an important approach, but there are limitations to it, which is why it's not the only approach. I don't know if anything else you'd add, Bruce, beyond those 3 to 4 we've mentioned so far.
Well, I do think that, you know, sort of what you said about looking at the biology first, I think the opposite thing happens where people look at the I would call the biology of the drug first.
Yes.
And then they go and look and see what else. Now that I know this drug, you know, sometimes you find a drug like the one you repurpose, sirolimus, also called rapamycin, and mTOR, the target is the mammalian target of rapamycin. We knew what it did and where it hit before we knew what it did. But once we figured it out, then there were all sorts of ways to think about repurposing that. So in both directions, looking at the biology, either disease or drug, can be really helpful.
That's a great point. That's a really good point.
Great. Thank you both for that. We are starting to get some questions. I'm going to ask one more kind of from that comes to mind that's more for general audience. What are what are some of the additional tools? I'm sorry, actually, we were just kind of going through that. One of the most important things for us to remember too is the role of the patients in this. So how can patients or researchers be stewards in this process and ensure that the research and the data aren't siloed? Or how can they kind of penetrate this world and be a collaborative partner in it?
Yeah, I think both David and I would say that trying to do drug repurposing without being patient-centric will almost always fail. At Helix, and when we were at Cures Within Reach, we were always making sure we had patient input, and it's just really critical. I see Blair, my friend Blair has joined. I remember when I met Blair and her partner Maria from Trend Community, we were talking about Prader-Willi syndrome, and I knew a little bit about this developmental disease. But had we started working on that, we probably would have worked on some of the neurodevelopment issues. But what I heard from Blair and Maria was the most important thing was making sure the kids could sleep, because they have sleep disorder. And when they don't sleep, the parents don't sleep, and then everybody's way less functional. And so you need to get the patient voice in there. Christian, you mentioned about the models and the data. I think these days, a lot of groups have realized that the fastest way to move things towards patients is for them to keep control, especially if they're funding all of the research. They're funding the basic research, they're gathering the data, they're supporting the registries and the natural history, they're building the models, cellular. The more they can control who owns that, who structures it, who gets access to it, the faster they can get more people working on it. And at our company, there are some relationships we have where it takes us between 6 and 9 months to negotiate and getting access to a model. That's way too long for a patient group. Other places where the models are at a contract research organization, and in 2 weeks we have the document signed and we're up and running. And that's often dependent, because the patient groups fund the early work, how that gets there. So we need their input, and we need their sort of control of things to make it easier for other groups. David, what do you want to add to that?
Yeah, I fully agree. The only thing I would say is that, and that's a really important perspective from a drug development side, is let's get patient input. And I would say if you were on the patient advocacy side, and just as Bruce said, make sure that you control it. I would also say if you're on the patient advocacy side, not only should you control the data, but make sure that you have the key stakeholders from the other aspects of the ecosystem that are deeply embedded within what you're doing. I think that sometimes, myself included, will focus on patient-driven research. And I think it needs to be patient-focused, but I think it needs to be community-driven, and it needs to, and not just like kind of in words, it needs to be truly patients, physicians, researchers, caregivers of patients, pharmaceutical companies, really as a unified front. And that's not always easy. Yesterday, Bruce, you and I were talking about, even within a given rare disease, there can often not be a unified front. And so first off, within the disease, there needs to be a fully unified front among patients. That's got to be kind of foundational step number one. And of course, we both recognize that there are reasons that there are sometimes, issues within communities, but that should be kind of goal number one, is get harmony within your community, and then make sure you bring on your team members, because it really needs to be, it can't be us versus them. It's got to be physicians, researchers, patients, advocates, all working together.
That's a great point. I think that came up a lot. David helped us a lot with our data DIY series, where we were teaching a lot of these fundamentals throughout the program. But one of the key things that we talked about was that strategy, having a strategy that's really inclusive and really is built by everybody involved. Thank you for that. Question from the audience. What criteria, and this is kind of a strategic question that involves repurposing, what criteria do you take into consideration to determine if a condition is best suited for drug repurposing as compared to an ASO or something, another approach?
Well, I can give the sort of Helix biotech answer. So for us, we need a number of things. So first of all, we need enough patients to do clinical work. So we look at the prevalence, both from can we mount a clinical trial if we need to, and are the patients going to be able to participate? And also, as a commercial company, is there a commercial viability to this potential asset? If we think there is, then we look and see what kind of data is out there in this disease. So we look for transcriptomic data, metabolomic data, proteomic data, and other kinds of things. Each one of these is a data point in our AI knowledge base. And the more data points we have, the more robust our predictions can be. Then we need models to validate those predictions. So cellular and animal models that represent the disease as much as possible. And then we also need to have clinical endpoints that we can use in a clinical trial in order to show whether a therapy is working. I mean, we look at some diseases are very slow progressing where the clinical trial might be 5 to 7 years versus other diseases where you should get a signal within sometimes a week or 3 months or 6 months. So from a commercial standpoint, that's helpful in not having a 10-year phase 3 clinical trial. And then we also look at the market. If there's already lots of things going on in that particular disease, if 6 or 7 biotechs and pharmaceutical companies are in phase 3 for something, then does it make sense for anybody to start at in the preclinical stage? By the time you get to market, the problem might be well solved. So from a commercial standpoint, that's sort of what we think about. David may think about things in a slightly different way because it's less commercially oriented.
Yeah, I think those are really good points. I would say that I generally don't think, when I think about rare disease drug development, and when we talk about drug development, we don't care whether it's repurposed or new. We just want to develop drugs, right? And when we think about we need drugs to help patients, I try not to think about it in an either or, should we do repurposing? Should we do ASO? Should we do gene therapy? I really think it needs to be an and equation where drug repurposing, as we said earlier, sometimes it's the thing that you do when you're backed against the wall and you're running out of time. You just say like, is there something out there that could save me? And it may not save my life forever, but in my case, it'll maybe keep me alive long enough to get married to my wife. I just wanted to make it 5 more months. And if this drug could keep me alive for 5 more months, then that is the patient-centered, focused outcome that I wanted. And so I think that's a similar sort of feeling that I think a lot of us have with rare diseases is we're not necessarily demanding cures now forever. There are things that we need. And so when I decided to start taking sirolimus and we went down this really drug repurposing centric approach, I knew that I had weeks to months left. And so there was obviously no way that I could do new drug development. If I had a little bit more of a longer horizon, I probably would have been able to think about drug repurposing and new drug development in parallel. But we were very much, okay, this is a now solution. There is no tomorrow. But I think that probably the more objective approach is not to say, I'm just going to do repurposing and I'm going to throw out all this other stuff. I think the right approach is actually to do both together. And we're trying to add in a little bit more of this longer term thinking in Castleman disease, but we're a pretty short-term focused organization.
And I'll add to that, the ASOs and the gene therapies may help a lot of patients, but they're going to be incredibly expensive and all patients won't be able to afford them. And they won't, in a lot of diseases where there's already been damage that won't be mended, but might not continue, there's still going to be a lot of symptomatology that needs to be treated. Barbara Goodman's still on. They have a program called Regrow, which is thinking about repurposing grants for the rest of the world. There's a lot of places that aren't going to be able to afford ASOs for decades, and we still have patients there that are going to need some therapy. And so drug repurposing can be an affordable way to reduce symptoms and be disease modifying.
I'm also going to share from Scott Barberin from UCSF that phenotype-based drug screening, specifically in zebrafish for rare diseases, has also recently shown some success. So I'm going to share a Nature article from Scott.
Yeah, I think there's all sorts of phenotypic screening things. Recursion Pharma, who are friends of ours, have this amazing phenotypic screening platform where they can screen drugs on millions of cells and take pictures of them and use artificial intelligence to see what's going on. So there's lots and lots of ways to do drug repurposing.
Thank you. So coming back, there are a couple more questions that I'm gonna circle back to, but in the meantime, I had another question lined up for you. what are the ongoing challenges you see in continuing to gather the data in off-label? Is it mindset? Is it technical? You know, what do you think the biggest barriers are, especially in light of what we're seeing in COVID?
I might start with an anecdote, if that's okay. I'm sure some of you guys saw the Surgisphere studies that were published in the Lancet and New England Journal of Medicine. And I have to say, when I first saw the studies, I was kind of like shocked, like, oh my gosh, how did they figure out how to get all of this data in one place in weeks? I've spent the last 10 years of my life trying to get a fraction of that data, and it's, you know, I've lost all of my hair because of it. And I think that, you know, what we've learned with Surgisphere is it sounds like most of it was fake, or that maybe it was maliciously and inappropriately brought together. So I think that made me feel a little bit better that maybe I haven't just been, you know, banging my head against the wall going the wrong direction. I think that I've been banging my head against the wall just like so many others have. And that's that, unfortunately, as Bruce said earlier, somewhere around 20% of all drugs that are given to patients are given for an off-label use. It may not be a rare disease, and it may be an off-label but acceptable use. It may be aspirin for the primary prevention of stroke as opposed to secondary prevention of stroke. So it's possible that it's those sort of things. But within that 20%, there's also a lot of these N=1s, people like me taking sirolimus. And so you'd say, oh, wow, it's happening all the time. We all have electronic medical records. There must be a seamless way that all this information comes together in one place, and there is not. So each health system has its own locks around the data within that health system. When data are shared, there are limitations, expense-related limitations. We've tried to get data on Castleman's patients to come from different institutions. It costs $20,000 per institution to set up the pipeline, and that's just the beginning. That's just kind of to say hello. And so in rare disease, that's unfeasible because you might need hundreds of health systems to accumulate 1,000 patients. And so there's systemic issues. And so what we've tried to do to solve that in Castleman disease, and solve is probably a strong word, but to address that is to, we created a natural history registry where any patient in the world can consent themselves online to join our natural history registry. And when they do so, they give us permission to reach out to that hospital to get their paper-based medical records. And we get those medical records sent to UPenn, where we have 3 full-time people who go line by line through every single page of every medical record, and they copy and paste into our database. It's incredibly time-consuming, very expensive, very resource-intensive. But where we are now 3 years into it is we have between 3,000 and 4,000 data points per patient on over 300 patients. And so we have a tremendous amount of data for drug repurposing. That's an expensive, time-consuming, slow process. But that, in my opinion, is one of the ways to do this, because unfortunately there aren't clear ways for it to happen seamlessly like Surgisphere.
Yeah, and I would add to that, there are some companies out there that are trying to accumulate that. We're looking at one called Trinetics right now, and we did some early work with them around some particular diseases that we're interested in at Helix, and found lots of patients. So, you know, the frustration that David has around Castleman's, there are some people out there trying to solve it. There's also some institutions, I think, that are doing some great work, like at Vanderbilt University, where they've been saving patient samples. Like if somebody takes your blood and they only use 1/3 of the vial, before they threw it out, they genotyped the rest. And then they de-identified it, but they connected it to any medical records there. And so they have this huge database that they use to find evidence of repurposing. And so I think there's some effort, Christian. It isn't where we need it to be, but you know, my hope is 5 to 8 years from now, when we're doing one of these global gene things, there have been some huge breakthroughs in using the information in electronic health records and other places so that we can more seamlessly make plans to repurpose.
That's
I'm glad that we have your optimistic perspective. We need to balance it with my jaded perspective. Thank you for that.
Well, I think I sounds like what's now and what's next. You know, that's
right.
And hopefully that's what we're talking about. And you know, at Global Genes, we are continually building up these networks and hopefully supporting the institutions that are going to encourage less publish or perish and more publish for patients, and keeping that in mind. Now, it does beg a question, Bruce, what you were saying. And David, you were mentioning it too, 3 to 4,000 maybe, was it data points or
Per patient, yeah.
per patient. So a big part of AI's role in this is the quality of the data. How important and what can, how important is it, and how can patients and patient advocacy groups mitigate the risks of uneven or disparate data?
Well, it's critically important, Christian, as you said, garbage in, garbage out in the world of computers, because computers are not, they're not using scientific knowledge to sift through and make subjective decisions about stuff. It's just objective. If you tell it to connect sirolimus with Castleman's disease, it'll go do that everywhere. And if somebody has the wrong drug name, if they spell it wrong, you're going to miss it. And so you really need to be careful about it. I do think, it isn't always the quantity of the data that's important. The quality of the data in AI oftentimes is much more important. So we'll find a well-kept small database of patients at a particular hospital, and that will be enough information for our AI to use. So making sure people are careful about curating the data. For us, no data gets into the AI without somebody looking at it. So much like David's group, reading line by line, whenever we get in data packages, somebody's looking at that to make sure that people follow the instructions, is everything lined up. But it's a big issue, the quality of the data.
A big issue remains as well on the topic, I'm going to throw a little bit of a curveball here, but on the topic of inclusivity and diversity in research. I wonder what's your experience been in terms of the accessibility and the quality of patients' data and patients' records when it comes to, or even access to them if they're not being seen at a center of excellence, at a Mayo Clinic, and what's the future hold for the ability to bring much more medicine and much more disease understanding forward with by being more inclusive with our community health systems and especially clinics in underserved communities.
I'm glad you brought that up, Christian. Certainly, I think many of us in science and outside of science are concerned about, but also committed to figuring out ways to improve inclusivity within medicine. I think that all of us in the rare disease space know that rare diseases do not discriminate. They affect everyone, no matter what your background is. And unfortunately, the health system is not always non-discriminatory. And so I think that there are ways that we can improve our health system. I think that, as you said, a lot of it has to do with resource accessibility. The registry that I mentioned earlier, thankfully, because we're getting paper-based medical records, we've not run into a clinic or a health system that can't send us at least paper-based medical records. I suspect if we were asking for HL7 formatted files that we might run into some more challenges, but thus far we have not. One thing I do want to offer to those attending is feel free to email me if you're interested in having the protocol that I mentioned that we use. I'm happy to share it with anyone and everyone, and just like you, Christian, committed to thinking through ways to improve inclusivity within science.
Yeah. And I would say, I think it's an issue that varies from country to country. I work for a company that's in the UK, and for most rare diseases over there is a center of excellence. And luckily, the country is small enough that all patients can, get there, and the government supports them getting there. So there's differences between there and what we have here in the US, where you might be 1,200 miles from a center of excellence. And for a lot of patients, that's rare disease patients, way too long a distance to travel. Christian, you asked a question about data. And one of the things that I think is really interesting and challenging about rare diseases is how heterogeneous they are. I mean, David said earlier, you expect something like Castleman's disease. All right, everybody has it. It's the same. Sirolimus should work for all of them. And it only works for a third. And we're not sure why. And so one of the extra challenges, I think, in the rare disease space is you're already starting with difficult situations with a small patient population. And as you get to the clinical stage, figuring out how to separate the patients that are likely to have a good result from the therapy versus patients that are not can be a real challenge as we plan for these kinds of things. So you asked me earlier, what do we look for? The more homogeneous a rare disease is, the more likely we are to be able to drive some kind of a repurposing therapy that helps the significant number of patients.
Very helpful. Thank you. So what excites you at this moment when it comes to the possibilities? We talked about what's next, and you've talked about, David, with the need to hold on to the sense of urgency that we've seen from COVID. Where do you think we're going, and what are you looking most forward to?
So I think there's maybe three things that I would lead with that I've observed from COVID that do excite me when I think about rare disease. So the first is that the incredible amount of open data sharing that we've seen with COVID. I think that we can prove that this can happen, this can be done, we can do it. And if people argue and say, well, it's just because it was an urgent pandemic, I think all of us can fight back and say, my child, this is urgent. For everyone with these rare diseases, this is urgent. So I think that now that the door's been open, I think we need to kind of rush the door when it comes to open data. I think second is around research coordination. There have been a number of efforts to bring together public and private partners to actually coordinate and track what research is being done, what trials are being done, who's doing it, to try to add some structure into research, which is typically a pretty random process. We found in Castleman disease that adding structure can actually help a lot. If you can provide the tracks, the train can go a lot more quickly. So I'd like to see more coordination in rare disease in a similar vein. And then the last is around drug repurposing, which obviously we've been talking about for this full hour. But I do think that the fact that we have been able to look at so many compounds and the fact that, as I mentioned, over 160 different drugs have been given to humans with COVID-19, we've proven that we can move really quickly from this is a good idea to this is in a patient. And I'd like to see us have a similar sort of urgency. And I should say, you should move urgently, but you need to also be very smart about how you interpret the data. And just because it works in a dish doesn't mean it's going to work in humans. So being rational with drug repurposing.
And I would say that the top things that have me hopeful are, I think there are lots of groups around the world that are starting to think globally about drug repurposing in the right places, like Cures Within Reach, like Open Source Pharma Foundation, and some others, Anti-Cancer Fund, all thinking really hard about how repurposing can help and getting a really strong foothold and making connections with each other. Second thing is there are groups out there that are working on new economic incentives for repurposing. And if we could just create some sensible economic incentives, I think the world of drug repurposing would slide right up into new chemical entity development, because it is faster, safer, cheaper to do. And if you can give somebody a reason for putting their money there instead of a new chemical entity, people will do that. The success ratio in repurposing is higher than in new chemical entities. So if you can just make money at it, people will do it. And then the last thing that I'm very optimistic about is that regulators are thinking about how we can simplify the route to clinical approval in rare diseases. Can we do distributive clinical trials? Can we use real world evidence? Can we do some things that help the wheel-bound patient, not have to go 6 times, 300 miles to a medical center to get treated? Are there ways of us doing this? So between, wearables and technology, I'm hoping all this stuff in the next 5 to 8 years really starts to come together. And when we're having these meetings, we look at research in a much different way, and the regulators do too.
Great. Thank you. I've got one last question for you that's a bit more of a walk me through it kind of case. One of our attendees is saying they're currently in a clinical trial for a specific disease, and this drug that she's in trial for, I believe it's a repurposing case. It's showing promise in another disease like ALS. Should they be reaching out to that ALS community about a partnership, or how would they go about engaging another community that's already actively in that kind of research?
Quick clarifying question. When you say showing promise, does that mean there's a trial of that drug in ALS, or does that mean that it's showing promise in in the lab?
I did not have that detail as to whether
Okay.
or not it's already in the field or not, but if we
Yeah.
wait one moment.
So it could be in the lab, it could be off-label anecdotal
Yeah.
information literature, or it could be in a clinical trial. So
Yep.
we sort of need to think about all three of those things.
And we've got another question coming in as well. So, oh, it's already in trial. Sorry.
In
Okay,
trial.
already
Yes.
in trial.
Yes.
So Bruce, I think you and I would probably say a similar thing, and I'll give a first crack at it. And that's that absolutely, I would reach out to the ALS community. I think that we talked earlier, you need unity within your disease, but I think we can also get strength from unity across diseases. And especially when it comes to dealing with the pharmaceutical company, there are some incredible pharmaceutical companies and executives I've dealt with. There are also some challenging pharmaceutical companies I've dealt with. And so if you can have multiple disease groups having a unified front in your engagements with pharma, I think that's only going to be positive for you.
Yeah, I would say that's true. I would, you know, if it was me, I would start to look at the protocols and the repurposing and see how similar it is and where there's an overlap. I try and get myself really smart so that when I go to talk to this other group or the key opinion leaders that are helping that other group or the clinical trial managers for that other trial, I want to make sure that I understand what to ask of them. And, you know, also to think of if this is a generic drug, that's different than if it's a proprietary drug. Because if it's a proprietary drug, you're going to be also needing to engage with that pharmaceutical company. If it's a generic drug or a combination of generic drugs, then maybe you can find an ally somewhere in the generic drug space that could, you know, that could work with you.
And there may be a partner organization in the other disease, in this case ALS, where they might want to help to co-sponsor the work. And there could be some cost savings from doing these things together. So I totally agree with everything Bruce said.
How does a generic change the dynamics versus something that's still on patent?
Yeah, I mean, the biggest thing is just that you're going to have a lot less luck getting any funding from a generic company and even from the initial manufacturer than if it's still on patent. Bruce?
Yeah, I agree. It's interesting when Cures Within Reach first started repurposing, we couldn't get pharmaceutical companies to support this in any way. They were always afraid that a drug they had for one disease would get tested in another disease and some side effect would get noticed or a patient would die and suddenly their blockbuster drug would be tainted. And now, you know, so many of them are thinking of indication expansion as soon as a drug hits the market. And interestingly, we didn't really talk about this, but you're starting to see drugs come out where they start in the rare disease and get some accelerated approval and some tax kinds of things, knowing that there's a more common disease. This happens often in the oncology space where, you know, you see as soon as a drug gets approved that there's clinical trials set up for, you know, dozens of other kinds of oncology diseases. So just have to be careful about that.
We are up against time at the moment. Hi, Blair. I knew I had a feeling you were going to have a question right here as we come up to the end. We are at end of session, but Bruce and David have been very generous, and they're going to make themselves available if there are any remaining questions. We'll make sure that we log them. We get back to you, and we'll put you in touch for anything specific to each of them.
Yeah, and I want to say, Barbara put in the chat from Cures Within Reach. I mean, she has just a tremendous amount of information there that could be valuable to anybody from the work that they do at Cures Within Reach. So, you know, reach out to her or Claire Thibodeau from Cures Within Reach, and they're glad to help.
Thank you so much for that. Thank you, everybody. The next sessions are accessible back through the auditorium and in the agenda there. So really appreciate everyone's time. Thank you again, David and Bruce. Wonderful job yet again. And go back and enjoy the rest of the afternoon.
Thanks, Christian. Thanks, David.
Thanks, Bruce.
Thanks, everybody. Bye.
Bruce, so what are we going to do tomorrow? Do we need to get on another Zoom tomorrow and keep this going and do it over again?
Sure.
I don't know what I'm going to do tomorrow afternoon.
It's Saturday, you know. So my significant other, Amy, and her daughter are doing the first venturing out. They're driving to Minnesota from the Chicago area where Amy's family lives, and they're going to stay up at the family lake house for 2 weeks.
Nice.
I'm going to stay home and take care of the cats. I'm in the vulnerable population age, so you know I got to take care of myself. But yeah, so anytime you want to get on for Zoom for the next 2 weeks, I'm free. I'm bachelor.
Deal.
So,
Deal. Oh, that's
Be
awesome.
safe, you guys.
yeah, thank you, Christian.
Thanks
I know you got to go and do
Christian.
so.
Always great to be on it. And Christian, I'll see you in like an hour.
Yeah. Yep.
Okay,
All right.
I'll see you guys
Okay.
there too. Bye.
Bye.